Okay, I'm going to wait 2 minutes. Yay! I just want to make sure the baby didn't miss the intro. Okay. All right, well, I'm going to go ahead and introduce you because I want you to get started, because I'm interested to hear what you have to say. This is Srinivasa Chakravarthy, whom I knew when I was a newly minted assistant professor at Baylor College of Medicine. I would say he did a postdoc with me, but I probably learned more from him than he learned from me, which is almost everyone that works with me would say that. He did his undergraduate degree at IIT Madras. He's a full professor at IIT Madras now. But after that undergraduate degree, went to UT Austin and worked with a person called Joydeep Ghosh, whom I knew at the time, and I don't remember why I knew him. I think we intersected a lot. Who swore up and down about you, and so Srini came to the first incarnation of my lab. I have no idea what that was like. He went back to IIT Madras and rose extremely quickly through the ranks. I think his CV has been circulated, and I think you'll hear today, I'll be surprised if this isn't true today. He's worked in a broad range of areas. I think he probably concurrently works in 6 areas at any one time. He has a real interest in the basal ganglia and what it does and how it could function and dysfunction in disease. He's worked in reinforcement learning models, both in the basal ganglia and in other aspects of things. When he came to my lab, he'd worked on complex valued neural networks, for those of you who know what that may or may not be. Everyone the feedback today has been that he's a person of broad interest and interesting ideas, and so with that, you're now not going to disappoint us, are you? Okay, good. It's nice to have you here.
Thank you, Reid. Thank you for inviting me here. Thanks to Christine for all the perfect arrangements, and it was really great.
Turn your mic on. And I think are we streaming? Is this streaming? Okay, so there may be other audiences, so don't
can you all hear me in the back? Yeah, and thanks to all you guys for coming here today. I just want to say I did 1 year postdoc at Reid's lab, and I just want to say one sentence about what I learned there. The first thing what Reid asked me to do when I joined is read Candle and Schwartz cover to cover, because I didn't know diddly about brain at that time, and so that's what I've done in the very first month. It was a great experience. But more importantly, more than the technical stuff, it's the inspiration that I got in that 1 year. In fact, what I learned there is doing science is not like doing a job, right, for your livelihood and for your pension and all that. He says it's like going for a war. When he used to see his battles with fights with Critic and all that was going on. So I mean that's how a scientist is supposed to work. So when I learned, that was really got into me. I hope so, a little bit of that. So I'm really thankful to him. I think I really owe a lot to him. So one thing I learned there is just at that time he was talking about this connection between TD error and dopamine signal in ventral tegmental area, right? In a very nice interpretation of Schultz experiments, and that has opened up the floodgates to application of concepts from reinforcement learning to function of basal ganglia. So when I went back to India, there's a little break after my postdoc. I did all sorts of things, and then around 2004, very fortuitously with some students who showed interest in that, I got back into basal ganglia. And we went off in a tangent in a very different direction. Initially, we didn't even know what we were doing. We just randomly trying different things. Things started falling in place, and we found that we have quite a different model of basal ganglia, which is quite different from the mainstream thing on that in that area, and it seems to explain a lot of things. So that's what I'm going to talk about. So I just want to give a little bit of my what Reid used to call a shtick. Right? What's your story? Okay. So so I think in spite of all the grand neuroscience methods that are being developed and huge number of models that are being floated around these days, I think we are still in a very primitive stage in the sense we have a lot of thumb rules for doing lots of things and tiny little models to explain a lot of phenomena, but we don't have this great integrative theories. Theory, we know the cycle, right? Experiment theory, and then new data, new theory, and so on and so forth. But what we have very often in neuroscience is this kind of a curve-fitting kind of models. A model that explains one thing and doesn't have any comment on other things. Whereas what we need is something like very deep, right, principles and a concept of principles. Because a good theory theories are meant to be universal. like theory would explain a falling apple and a falling moon and tides, and a biologist would say an apple is different from a moon and is different from a ocean tide, but you need to have theory which explains all of that. So that's the kind of thing we would like to achieve in basal ganglia. Obviously, we haven't reached there, and it's a long way to go, but that's a kind of an aspiration to get there and understand basal ganglia's manifold functions. I mean, Marsden, the famous British neurologist, talks of the mysterious functions of basal ganglia. So, it's mysterious because it's so many-sided, right? And everything, if you look at Parkinson's, which is impairment of basal ganglia, you see symptoms going all the way from apathy and insomnia to constipation. I mean, how can the same circuit have such wide-ranging symptoms? So that's the question. So I don't need to tell you guys about basal ganglia's anatomy, but it's an important subcortical circuit with all these nuclei. I'm sure we all know about this. A simple block diagram of all these nuclei would look like this. So you have signals coming from the cortex going through the basal, this whole thing is the basal ganglia, and going back to cortex by a thalamus. And then there is this important structure called the substantia nigra, which projects to actually pretty much all nuclei in the basal ganglia. And then the input port to the cortical inputs is a striatum. There are two output ports, GPA and SNR. And there's a shorter route from striatum to outputs, which is a direct pathway, and there's a longer route via GPE and STN and GPI. That's an indirect pathway. Okay, this is a standard anatomical textbook description of basal ganglia. Now, basal ganglia functions are so wide-ranging. In fact, basal ganglia impairments are seen in the consequences are seen in all the four major domains of brain function: motor, cognitive, affective, and even autonomic. So here is a short list of functions, but this is no way a complete list. There are a lot more functions. So people have been trying to model different functions of basal ganglia. Some of the earliest pioneering models are from Reed and Schulz, and that line of work, like reward-based functions of basal ganglia. And then the sequence generation stuff from Burns and Sejnowski, action gating and action selection, and working memory, timing, and dimensionality reduction. So point is, very often you see that not only with basal ganglia, for most neural structures, you have a kind of half a dozen schools of thought, right? Half a dozen camps. And each camp pursues and describes that structure in their own unique way, and there's hardly any consensus from one camp to another. In fact, if you look at the book from 90s on information processing models of basal ganglia from MIT Press, right? It's written by all the well-known names in the field, and you have like half a dozen different perspectives, and so that is a problem with basal ganglia or pretty much every other structure. It's like this blind monks trying to figure out what an elephant is like. So, okay, so if you understand basal ganglia, you can understand PD, and PD is a neurodegenerative disorder, loss of cells in the substantia nigra. But that's not the only story. I mean, PD actually is a very complex problem with PD like this Parkinsonian plus and all these things. Okay, and then there's the cardinal symptoms: tremor, rigidity, posture, and bradykinesia. And you have also all these motor symptoms. So what is very intriguing is loss of this tiny little nucleus can give rise to pretty much everything that you do in the voluntary movement area, right? And from speech, reaching, handwriting, eye movements, posture. So how can this happen? So this is a question we are trying to address using computational models, and come up with a mechanistic cause and effect model of why loss of cells in this tiny little part of the brain gives rise to such wide-ranging, at least motor symptoms, not to speak of other things in the autonomic and affective area. So let us go back to the classical picture, what you would kind of stuff that you read in Kandel and Schwartz, and then from there let us see what is wrong with it, and let's try to see if we can make any modifications to that. So classical picture goes something like this. This is Albin et al. Kind of stuff. So you have the direct pathway and indirect pathway. If you activate the direct pathway, right, you have facilitation of movement, and if you activate indirect pathway, you have inhibition of movement. And this is often also described or accounted for, argued in terms of number of neurotransmitter stages, excitatory, inhibitory. So you have two inhibitory stages, GABA stages from here to here and here to here. So effectively, you are disinhibiting thalamus and therefore cortex if you activate the direct pathway. Whereas on the indirect pathway side, there is an odd number of inhibitories, 1, 2, 3. So therefore, effectively, it is inhibitory. So these are the kinds of arguments that are proposed or preferred to explain this kind of simple duality between direct and indirect pathways. So therefore, this pathway is called the GO pathway, and this pathway is called the NOGO pathway. And interestingly, dopamine is released by SNC here to striatum. And interestingly, the action of dopamine here acts like a switch which can control the transmission of the corticostriatal information directly via the direct pathway or through the longer route, because of the kind of neurochemistry that happens in here. I'm not going to go into these details at this point. So basically, for high dopamine, delta naught here is like dopamine. For high dopamine, you have GO. For low dopamine, you have NOGO. This is a kind of a picture that was, so for high dopamine, you have a hyperkinetic condition, for low dopamine you have hypokinetic, the kind of thing that you read, right? But that's a very simplified story because you feel the need for expansion if you look at many other facets of dopamine, for example. The dopamine is called the pleasure chemical of the brain because it's triggered by brain stimulation of the reward centers, right, psychomotor stimulants, opiates, and even food. And dopamine agonists actually mess up this experience and kind of spoil the fun. And also presentation of beautiful faces, images of lovers, cash rewards, you guys work with this. All these things trigger dopamine reward system. So dopamine is a part of a big reward network, right, and whose activities match very well with what we know as reward and pleasure and things like that. So Schul's early experiments, seminal experiments, kind of confirm this, but actually tell you something more than this. So in this experiment recorded from VTA, as you probably all know, right, there's a conditioning stimulus, and after a delay there is a reward. Actually, monkey can reach out into a box, a closed box, and grab a piece of apple which is stuck to a wire. And at this point, initially in the early trials, the VTA cells fire only at the time of reward, but after some pairing of the conditioned stimulus and the reward, the cells start firing right at the time of the conditioned stimulus, as though the firing increase indicates a prediction of reward, predicted reward, right? Because CS predicts reward. But in the third case, the CS is given, but the reward is omitted because the box was made empty. Kind of animal was cheated, and in that case, there's a depression of dopamine firing. It is as though the animal is disappointed. So basically, what this says is the firing pattern of these cells indicates not just reward or predicted reward, but the difference between the predicted reward and actual reward. Right? So this matches well with a quantity called temporal difference error in reinforcement learning literature. So in fact, with that, the whole theory of reinforcement learning, which is originally inspired by animal psychology and instrumental conditioning, right, has been progressively applied to basal ganglia function, and a lot of work has happened on those lines. So in essence, the reinforcement learning works like this. So if there is an agent or an animal which maps a stimulus onto a response, right, and for a given stimulus, if the response leads to reward, then you strengthen or reinforce that connection. If for a given stimulus, if the response leads to punishment, then you weaken or attenuate that connection. So this is, in a sense, the reinforcement learning. Now, if you look at the basal ganglia's anatomical position, it is well placed, right, to do exactly that. Because if you look at this box in the previous slide, right, this box has three things coming together. The stimulus, there is response, and then the reward information. And in the basal ganglia, there is stimulus information comes from sensory cortex, and response information comes from the motor cortex. And the reward information, or actually the TDR, comes from the SNC. So it is well placed to perform that kind of a task. So it combines all this and makes a correction, right? Prescribes a correction to the motor and executive areas of the cortex. So it is doing some kind of a reinforcement learning. It is some kind of an RL engine. So it's a reasonable thing to kind of assume as a first cut. Now let us look at these concepts more closely with a simple toy schematic. So here is an animal which is looking at two choices, yellow and red. And when yellow gives you reward and red gives you punishment, but the animal doesn't know that initially. So initially it has kind of equal weightages to both these choices, and then it presses yellow, it gets reward. So the weightage for yellow goes up, right, and then it presses red, right, it gets punishment. So weightage for red goes up. So next time around it's going to choose yellow and try to avoid red because so these weightages that it learns, right, are like values, right, that it learns, and the values are now act like surrogates to reward or punishment. What it gets after the action is performed. Right. So the simple story is choose the ones which have high value, avoid the ones which have low value, right. But there is a third thing which is necessary to complete the picture, to complete your whole reinforcement learning picture, which is exploration. So let's take an example of going to your favorite restaurants. So the weekend, if you want to go to a restaurant, you will go to your favorite restaurant. You'll avoid the restaurant where you had bad experience. But that's not the whole story. If you want to optimize your choice of restaurants, because there are always new restaurants coming up in the city, and you would like to try out new restaurants, and that's called exploration. So if you want to be a complete and optimal RL engine, you should be able to do all three, not just the two. Right? So whereas the classical picture of basal ganglia only talks about go and no go, right? It doesn't really spend too much time on exploration. So to summarize, RL machinery involves all these concepts. There's a reward signal, there are values, then there's exploration, actions, and state. So if you think of basal ganglia as a comprehensive RL engine, right, what are the correlates of all these quantities. Now state comes from the sensory cortex. Actions you can say are performed by the motor cortex, which is the output. Now reward or TD error comes from SNC, and values are thought to be stored in the striatum because a lot of data from imaging from Doherty, and all these people, right? But the missing piece here is exploration. Where is exploration going on? Okay, so this is the classical picture, go and no go, and so thing is, where is the exploration? And so we propose that we propose that the indirect pathway is doing exploration, and the classical picture where indirect pathway is simply treated as a no-go pathway is not quite accurate, because one thing that picture misses is this feedback connection from STN to GPe. Now because of the feedback, the indirect pathway forms an excited inhibitory loop, right? And such a loop is well known in computational literature that is capable of producing complex dynamics. Okay, so your arguments in terms of odd number of inhibitory states is simply not correct because of this loop, which gives you a very special system, right? And so what we are saying is this indirect pathway does not only no-go, but it also does exploration. It is what gives the output of the basal ganglia some kind of exploratory drive, which will enable it to choose actions randomly. So I'll spend more time on that exactly what happens. So we proposed an expansion to the classical go-no-go picture. So it's consistent with the classical, but it says something more. So for high dopamine you have go, for low dopamine you have no-go. For intermediate levels of dopamine, this new regime emerges, right, which we call exploration. Okay, let's discuss this a little bit further. Now, if you look at the RL components and the neural subsets of RL, if you look at TD error, that's dopamine cell activity. Actor, okay, this subcortical, it is a dorsal striatum, and cortical, you can say it's motor cortex. For critic, subcortical substrate is ventral striatum. Cortical, it is orbitofrontal cortex. For explorer, people have found cortical substrate. There is anterior frontopolar cortex in the prefrontal, and intraparietal circles in the parietal. But there is no subcortical explorer. People haven't found anything. Okay, so how can everything be there in the cortex, but in subcortex? But how could this piece be left out? So to summarize, if you look at actor, critic, and explorer, all three are present cortically, but only these two are present subcortically. Explorer is missing. Okay, so does it exist, or should we not worry about it? Now here we are saying that STN-GPe is the explorer. Okay, so how do you justify that? In fact, what do we know about STN-GPe? What have people been saying about it in the past? So no-go pathway is a standard thing, but then people have said other things also, like Hikosaka, the well-known name in Saccard's literature. He says STN-GPe gives you focusing. So that's because the projections from striatum to GPI have wider receptive fields compared to the projections from striatum to GPI. So therefore, one is inhibitory, one is excitatory. The combination of two such receptive fields gives you kind of a Mexican heart type receptive field, and that will give you focusing. So, and then alternate activation of direct indirect pathway can give you sequencing. I mean, these are the kinds of arguments that have been presented, in these reviews. Then action selection, there's a big British group, Gurney, Prescott, Humphreys, and all these people. They talk about action selection aspects. Then switching again, switching from automated to kind of more voluntary decision making. When the switch occurs, again, STN neurons are found to be active in this Saccard study by Isoda Hikosaka. So I think several things have been said. Another thing I noticed is the importance given to the STN-GPe is somewhat tentative. Right? Sometimes it's there, sometimes it's not there. It's like a given very stepmotherly treatment. So now that's from experimental data. So what does the modeling guys say about it? So like I said, the STN-GPe forms an excited inhibitory loop, and this is a classic template, like archetypal template that can produce complex oscillations, and there may be many studies on that. So Gillies and these two papers have looked at simplified nonlinear limit cycle type models, whereas Turbin et al. Looked at a very detailed biophysical model. Both have, in fact, Turbin et al. Call the STN-GPe the pacemaker of the brain. It's some kind of a rhythm generator of the brain. So but the thing is, these models only focus on STN-GPe. They don't tell you what is the role of this system in the big picture in the decision-making operations of basal ganglia. Okay, now from experimental side, our question is there a subcortical explorer, and is it located inside the basal ganglia? Let's look at some more data. So Grillner et al., very old data from basal ganglia, they show that decorticated kittens show exploration. So if there is no explorer in the subcortex, how are they able to explore? What is the substrate? Now STN lesions cause perseverative behavior, Bones et al. right? That means if a lesion is in STN, the animal is going to choose the same action even though it's not rewarding. That's like destruction of exploration. So that means that's already indicating that STN has something to do with exploration in the subcortex. Also, people have found from electrophysiology, Bergman and Nini and all these people, that there is a reduced complexity of the STN dynamics, STN-GPe neural dynamics under conditions of dopamine deficiency, like in Parkinsonian condition, but there's not much change in the firing rate. So higher, as you see, the synchronized bursts in STN are a classic sign of a Parkinsonian condition, Parkinsonian dynamics of basal ganglia. So in terms of complexity, it means there's a loss of complexity. They're all firing together, so there's not much information in that. So this is probably a clue, another clue that the STN has a role in exploratory behavior. Okay, so I've just put the pieces together, and then let us now look at the model. So the first model, which is a very simple model, right, where all these intuitions are brought together, and we want to see how this exploratory behavior actually emerges out of the dynamics of the model. So in this simple model, we have striatum, then we have direct pathway, indirect pathway. There is GPe, STN. There is feed-forward feedback connections between GPe and STN. Then there's GPA and thalamus, and output goes. So we looked at a very simple problem initially. So we have just two inputs. There is A and B, two choices. These are like saliencies of some choices. You're choosing between yellow and red or whatever. So A is more salient than B. So normally speaking, you should be able to choose A and avoid B. Now we look at there's no learning in this initial model. I put in learning a bit later. So here I just vary the dopamine level from SNC to striatum, and I want to see the action, what action is selected as a very dopamine level. Okay, so let us look at that. Just a model setup. You have D1 medium spinal neurons and D2 medium spinal neurons in striatum. D1s project directly to GPI and D2s project to GPE. So all that we have taken care of. Okay. So the point is, some people think that computational models are all about taking a big neural network and training it to death, and so obviously you can't do that. You have to take as much data as possible and kind of carefully construct the model by looking at as many constraints from biology as possible. Then the STN-GPe model again has this feed-forward feedback connections, therefore it's able to produce oscillations, and you can play with the parameters, and you can produce chaotic dynamics here, or you can produce very regular waves, right? All that you can control in this. It's also consistent with a lot of other studies. I'm not going to all that in detail. So direct indirect pathway outputs meet in GPI, and then it goes to thalamus. In thalamus, we assume some kind of a simple competition going on, and then the winner is selected. Okay, so this is basically the model, very conservative, nothing too fancy. Now, if you do that, and then if you run the model and look at the model as a function of delta, delta is the dopamine, right? That's what comes from SNC to SNC to in this striatum. If you vary that, for high dopamine levels, which is like 1, the model selects the better action, the A. For very low dopamine, doesn't do anything, doesn't select, because the action is selected only when either of the outputs crosses a threshold. In fact, the output which first crosses the threshold is taken as the decision. What is called a race model in Saccard literature. So we just use that. So for low dopamine, nothing happens. High dopamine chooses the correct one. But for intermediate levels of dopamine, it does something very strange, and it comes automatically out of the model. We didn't put it by hand, right? It chooses A or B with some level of stochasticity. Now, all the equations in the model are completely deterministic. There's no noise term anywhere in the model, right? And in fact, whereas here, but in the output, you just get this. There's a little hill of exploration. So this is a probability of choosing the non-optimal choice, right? So what happens exactly? Let us go, and this schematic shows what exactly happens inside the network. If you look at high dopamine, this is a better input, and this is a better one, this is a worse one. And then for high dopamine, the D1 neurons are more active, so you just that selects the direct pathway, and then because of that, right, the action A is selected. For very low dopamine, everything is silent, and they are not able to cross the threshold in the output nucleus, so that there is no selection. But somewhere in between, both direct and indirect pathways are selected to some extent. So the STN-GPe, because of the oscillatory dynamics of these complex oscillations, it's able to give these random kicks to the output, and therefore in every trial for these intermediate levels of dopamine, you get a different output. So we are saying this is where exploration lies. And so some people, to justify exploration, they say that, oh, there's all sorts of noise in the brain and it's always there, and why worry about it, you take it. But that's not okay, because the exploration can be modulated. For example, in the model, if you vary a parameter epsilon s, which represents the lateral connectivity of neurons in the STN, you can modulate this exploration. So if for this low value you get a very high exploratory hill, but by increasing this, you can suppress that. So there's not much exploration; it will become quite deterministic. So it's quite a tunable kind of a behavior. So with this basic result from the model, we are proposing that there is the three regimes, go, no go. Between them there is an explore regime. We call this the GEN or a GEN policy. Okay, so far we haven't talked about learning. It's like you have basically a box. There's input and there's action selection in the output, and the dopamine is like a knob, right? As you turn this knob, how does action selection vary? Okay, so for high values of dopamine, you select the best one. For low, nothing. Okay, so now you want to close the loop and put in learning. So we are applying this to the standard action selection task, the n-bounded problem. So you close the loop, either from thalamus back to the cortex, or from cortex back into the striatum. Thalamus back to striatum or cortex back to striatum, right? And then your SNC acts like a TD error, and then we introduce one more parameter which we call the value difference. The TD error is RT plus gamma VT plus 1 minus VT. This value difference is simply VT plus 1 minus VT. Okay, so we felt the need for a quantity like that. So I'm not going into the kind of math of it, but if you train it on a standard n-bounded problem, it will just behave like your standard RL models, right, with the softmax principle or epsilon greedy, and it will with after some trials it will learn to pick the best action. Okay, so go to the pick the best action, and here you have epsilon greedy. In softmax you have beta, and the equivalent to that is epsilon s, which is the lateral connectivity in STN. Next, we extend this to continuous action spaces. So basically what you are doing is, when you have n-bounded, you have multiple actions, each action has a value, you pick the action which has the highest value. So you're kind of doing some sort of optimization, but over a bunch of discrete values. But if you extend that over continuous spaces, you're not picking from a bunch of discrete values, but you are doing hill climbing over the value landscape and going towards points which have higher values. So this whole model can be summarized and reduced to a very simple three rule set of equations. All right. So for example, how do I climb the value? So I go a step in this state space, in the action space, and because if this step gives me much higher value than before, then I continue going in the same direction because the previous direction was great. Now, or if I take a step and my value dips too much, then it's a very bad choice, very bad action. So next time around I can do one of two things. I can turn around and go exactly in the opposite direction, because that way value is going up. All right, I don't do anything. That's like no go. Now, if I take a step and my value doesn't change much, it's neither great nor not too bad, then I go off in a random direction. That's explore. So three regimes we talked about. When you apply the same model to a continuous action space, you get something like this. Okay, this is like a reduced, algorithmized version of the network dynamics. It's not something that I'm just making up and putting it by hand. So if you do that, then you get a very interesting optimization algorithm, which is coming out of this detailed network dynamics. It has some similarities to simulator annealing, but it's just interesting, that it looks like that. So to summarize, so that kind of sets up the basic model, right? So what we are saying is DP, direct pathway does exploration, and indirect pathway does direct pathway does exploitation, indirect pathway does exploration. And STN-GPe is like an explorer by virtue of its very complex dynamics, and you can produce chaotic activity in indirect pathway, and this is what is driving the exploration. In the Parkinsonian condition, that chaotic activity is lost, you have a collapse of complexity, right, in PD conditions. So therefore, you don't get very optimal exploration. And values computed in striatum. We have two dopamine signals. One is the standard TD error, and another what we call the value difference signal. Okay, and the GEN policy gives you basically a stochastic gradient ascent algorithm over the value function. So with this basic ingredient, you are able to explain a variety of functions, mostly motor functions, in spatial navigation, saccade generation, applied to also deep brain stimulation. So we wanted to explain PD speech, but we realized quickly that PD speech is quite common. Speech is very complicated. So as a first step towards that, we did some modeling of bird song. Birds also have a basal ganglia, although it's not a mammalian basal ganglia, but same concepts are kind of replicable. Then precision grip, that is how do you hold an object between your thumb and index finger, right, and lift it, and then risk-based decision making and so on and so forth. Okay, so all that has resulted in this book, which has just come out a couple of months ago. Okay, so I'm going to talk about 3 functions. One is simple reaching model, and then gait model, and then modeling build action. So reaching, we all know what reaching is, so I'm not going to spend too much time on that. So in this model, there is a simple 2-link arm, so an arm which can move in 2 dimensions, and there is a cortical loop. So in this version, we added a cortex to the basal ganglia, so we're gradually expanding, right, moving away from basal ganglia and adding more stuff to it. So the first input of the arm goes to what we call proprioceptive cortex. I mean something like a sensory cortex or some kind of somatosensory cortex. And then the proprioceptive cortex is trained by the muscle lengths and stuff like that. The proprioceptive feedback. And then output of this goes to a motor cortex. And we say that the prefrontal, which has the goal information, right, prescribes where to move. Right, this is my goal state. You move your hand to that goal state. So that information comes from prefrontal. And then the output of motor cortex goes to motor neurons on the spinal cord, and which controls the arm. Then the output of motor cortex and the proprioceptive cortex goes to the basal ganglia, and you go through this whole, right, all these loops and all these dynamics happen inside basal ganglia. Output of action selected, adds to the motor cortex. So motor cortex receives input from many sources. Basal ganglia output is one of those sources, which is influencing motor cortex. And we have a complicated scheme for training all this in a stage-wise fashion. So this is the workspace of the arm, okay, standard stuff. And so once we train the network, we see the activity in various areas evolving. So here is the red dot is the target, and then the arm is moving towards the target. In the early stages, it kind of moves around, kind of gropes around and seeking the target. But with practice, as you run this for a long time, it will just make this clean sweep right towards the target once it is trained.
Does it get reinitialized every time the red dot moves?
No, it just keeps on going.
So it learns.
Yeah, so here it's still learning, it's still seeking and learning. Okay. But with practice, it just goes towards it. So now in PD conditions, so PD we do, to simulate PD, we do two things. We suppress the dopamine signal because it is a dopamine deficient condition. And one more thing is we change a parameter in the STN-GPe to make the dynamics more regular, because that's also seen in PD condition. So these are 2 things we use to simulate PD. And when we vary this parameter in STN, right, in 2 different ways from the normal, what represents the mean normal. In one case, the arm, when you train it, right, it shows this kind of behavior, tremor. Okay, it's not reaching towards target, but just keeps shaking like that. The other case, it doesn't even move. It's like your akinetic rigidity, right? So in fact, PD patients come in 2 different flavors, right? You have tremor dominant and rigidity dominant. Sometimes both are simultaneously present, which makes it even more complicated. But what we found is if you vary one parameter in opposite directions, you get 2 opposing kind of symptoms. So, sorry.
So can you simulate injecting dopamine?
Yeah, you can.
For example, to undo the rigidity, or?
So thing is the 2 things. Dopamine reduction is a starting point, but the whole network continues to learn with that parameter, and that will mess up your cortex and connectivity and all that. So now if you just be adding dopamine, it's not going to fix the problem, because other things have already learned on top of this bad error signal. So I'll show some of the consequence of that in case of gait. Right, so we have to understand the complete dynamics before we plan a treatment. Simply replenishing dopamine won't work if it's already happened for too long. Okay, so all the standard stuff, like you have simple reaching speeds, right? Reaching velocity looks like this for controls, and speeds are lesser for PD because bradykinesia, and then the peak velocity is also lesser in case of PD. This is all from this study, and we are able to explain this data. Time to peak is higher in PD, and that movement time is also higher in PD. So another thing we notice is that the trajectory path variability.
So are these all bradykinesia explained?
Yeah.
So anything that would slow it down would.
Sorry?
Anything that would slow down
Yeah.
Planning or execution or valuing
Yeah.
of a movement
Yeah.
would account for these data.
Maybe. Yes.
Okay.
Yes. Yeah. Okay. So you see.
Can you answer another question?
Yeah.
Never mind. I'll wait. Go ahead.
Okay.
Sorry.
So we see higher path variability in case of in the PD condition compared to normals. Okay. And so basically this picture summarizes it. By varying some parameters in the model, the y-axis is distance to the target. So in normal reaching, your target to the distance to the target is 0, becomes 0 very quickly. But in pathological reaching, this oscillates, or it's frozen, doesn't really move towards target. So this is what this graph represents. Okay, so let us go to freezing of gait. Now, PD people have very interesting gait deficits. For example, they walk slowly, there is the step size is smaller, and so on. In addition, they show a symptom called freezing of gait. So they are walking in, suddenly they kind of are stuck in their tracks and they just go like this. I mean, you might have seen videos on YouTube, right? So why does it happen? Is there a theory of that? So we applied the same model to PD freezing to PD gait, and we were able to explain why freezing can occur. So the first study that we simulated is from the two studies, Almeida and Lebold and et al. And Cowie et al. So in this study they have a doorway, and then the subject is asked to go through the doorway, normals and PD patients. The doorway can be under narrow, wide, medium, or wide. Normal people just go through the doorway, but PD subjects slow down a little before the doorway, right, about a meter before the doorway. So why does that slowing occur? And that slowing actually precipitates into freezing in case of freezers, PD freezers, because not all people have freezing behavior. So it is set up as a reinforcement learning problem like this. So here this bar is the doorway and this circle is like the agent. I mean, there's a lot of wobble in the motion just to make the problem going. And the idea is if it goes through the doorway without straight, without bumping to the sides, it gets reward. If it bumps to the sides, it gets punishment. Okay, and then the input to the agent is the visual appearance of the doorway. So you have doorway in the center and there's walls on the sides. You kind of code that in a simple vector and give that to the agent, and then you give the reward and you train it. So here is the scheme for the network. I'm not going to details of the model, but it's a very simple formulation. If you do that, and if you, so there's a normal model, and then for the PD model, we do exactly the same thing as what you have done before. You suppress the dopamine signal and then change the other parameters like in STN and so on. If you do that, we found that the model also shows slowing before the doorway, and just like the human subjects. Okay, so that's interesting behavioral readout, but what happens in the model? Why is it showing different behaviors? Now we looked at the value profile. So this is done by my PhD student Vignesh. So if you look at the value profile, right, in the normal condition, the value climbs up towards the doorway, and that makes sense because if here is a doorway, I'm far from the doorway. Value indicates my probability of getting a reward ultimately, right? So if I'm far from the doorway, I'm not sure if I'm going to make it ultimately, so my value is low, far from the doorway. But as I come closer and closer, my bets are more and more I'm reliable, so I know that I can make it by just walking through the doorway. So value should keep going up towards the doorway, and once you cross the doorway, you already got the reward and no more value, so your value dips. But in PD conditions, we basically suppress the TD error signal because your dopamine is less, and with that suppressed error signal, we train the value function. Okay, so value function is going to be wrong, and that wrong value function ends up looking like this. It's actually dipping towards the doorway instead of a normal increase towards the doorway. And because of this, you see the freezing that you see in PD patients. So the same model is able to explain other data from the papers.
Wait, can you go back? Why is it, I'm asking the big question.
Yeah.
Why is it learning basically the inverse of the negative of that? Why is it learning that value as it approaches the door?
So
in the model,
the first one
once
is,
you remove the dopamine, and it has a hard time distinguishing value.
yeah, no,
Why does it learn this big hole?
Yeah, so the error signal that you have in PD condition is a wrong error signal. We suppress it. Suppose my error happens to be 5, right? In normal case, for a given situation, I make it 2 or 1 or something like that.
Why wouldn't it learn a small positive hump instead of a whole?
No, you're training.
That explanation seems like it would say the value function that will get learned is a small little positive hump approaching the doorway instead of the whole. I'm just not understanding how the elements.
I don't know if you can verbalize the result in simple, I don't know. Because the model is, basically with a wrong error, if you train value function, it's going to look very different from this. And what we found is, right, something like opposite. And
Your
yeah.
PD model, when you presented it with this depiction of this problem, learns that.
Yeah, yeah,
Okay.
yeah, yes.
Presumably the way you were showing it, if they have more sort of abnormal gait,
Yeah.
that they would be more likely to get a punishment, or is it just a lack of reward
Yeah.
when not going through the door, hitting the door edges?
Yeah, so
Does
the,
that have anything to do with the more likely to hit a door edge and the negative valuation?
so, yeah, so like I said, you get reward when you go through without bumping to the sides, and also you get punishment when you go very far up the track, okay, laterally, to left or right. So once you set it up like that, so your reduced value now starts looking like punishment. Your reduced delta will start looking like looking very close to the punishment signal. So I don't know if I had answered your question. I mean,
In a real sense, I was just thinking they're more likely to run into the doorway because of the irregularity of the gait.
yeah.
Would be a punishment.
No, so the thing is, they understand that the doorway is punishment, but therefore they actually slow down. So I think they're hitting the no-go regime. So if they're in the explore regime, they would randomly bump into something. But it's very bad, very low dopamine levels. The signal is clamped at a very low level. They would actually freeze. In fact, the extreme case of this is what we see as freezing. Okay, so we explained the changes in stride length. These guys looked at, Cowie et al. Study looks at wide door, medium door, and narrow door, and we see all these changes between controls, PD on case. PD on is better than PD off. We see the same trends. And in the Almeida and Lebold case, they look at, again, wide, medium, narrow, but they look at controls, PD non-freezers and freezers. Okay? Yeah, good. Yeah.
Neurologists, motor symptoms associated with PD, there are lots of them. One is this freezing or inability to start, are being treated with SSRIs in some subclasses.
Yeah.
How does that fit?
Yeah,
with
beautiful
what you're
question.
depicting it here?
So when we're trying to fit this data, right, obviously you have to tweak the model parameters and all that. So the parameters are, there is this
Would you have guessed that?
Oh, no,
Okay.
no, I won't have.
If you'd taken your model
Yeah.
and you had not heard this so-called folklore about the neurologist, would you have said, I need another parameter here?
No, okay, no, wait, no, we would have guessed that, but we wouldn't know what to do with it.
I see.
In the model, we have this sigma, which controls exploration hill, and gamma, which is discount factor, and then the delta offset value, which tells you how bad the disease is, how much I should crush the delta signal. We play with these three parameters until we get a good fit, right? And we found that to get a good fit, it's not enough to only suppress the dopamine. We also have to play with the sigma and the gamma. Gamma represents norepinephrine according to Doya, and sigma represents, serotonin according to Doya, and sigma represents exploration or norepinephrine according to Doya. So we felt the need. So the parameters are different between normals and PD freezers. And not only that, we could distinguish between PD freezers and non-freezers. But both have dopamine deficiencies. So what's different between the two? It comes in these two parameters. So in fact, to get a good profile of PD patient, you should look at all four deficiencies, or at least all three, right? Dopamine, serotonin, and norepinephrine. So in fact, this leads to a lot of interesting clinical applications, because if you can take the clinical data, like ask the guy to go through a doorway or whatever, and then measure this data, put it back into the model, and invert the data in terms of the model parameters, you have a very precise profile of the patient, right? And then you plan, how much serotonin medication to give, how much norepinephrine medication, how much L-DOPA. You think of the dosage combination.
Instead of having them walk, do you think that the PD patients would equally have a hard time moving a manipulandum-controlled ball on a screen? So they ought to start freezing near there. They ought to develop
Yeah.
And so you could do it in a screen.
We could do that. In fact, one other prediction that we have is,
I say that because there's a whole subclass of PD patients that have postural problems, unrelated to the initiation.
Yeah.
They can't stand up very well. They can't
Yeah.
stabilize very
Yeah.
well. But you would still predict, if I was moving something toward a goal, toward a doorway on a screen, that I ought to start slowing, and I should freeze at the doorway.
Yeah.
Even controlling something on a screen.
Right. Yes.
Predict that.
Yes. So if, suppose you are, you're buying tickets for a movie, you put your hand in the counter, right? Your hand should freeze at that point.
So we could do that in surgical suite while we recorded
Yeah. That's awesome.
directly.
Great. Yeah.
You would predict.
Yeah.
This
The
model would make distinct predictions here.
PD freezer should be have. Yeah. The hand should freeze. Yeah.
Hand should freeze.
Yeah. So, and similarly, they have higher coefficient of variation, and that this trends also can be seen. Okay, so basically, to summarize this, we are saying that the moral of this whole study is, PD gait is not a motor problem; it's a problem with space evaluation. So normally, when you walk, you are navigating in space, right? And you are getting geometric information about how far the targets are, how far the obstacles are, and so on. But your value system, the reward, basal ganglia, and dopamine, and all that, is evaluating how safe this space around me is. So if I walk in this direction, that is safe. So my value in this direction is going to be high. But if I walk in this direction, I'm going to bump into something and hurt myself. The value in this direction would be low. So the reward system, your visual and parietal, all these are modeling your space and telling you how to navigate. But the value system is telling you what is a safe path in this to navigate. Right. So and that is messed up in PD case because your delta, which is necessary to compute that, is messed up. Okay. So in fact, that gives rise to a whole concept of evaluating space. Right. For example, you can talk about spatial discounting, because in RL always talk about temporal discounting. Right. Whereas so it's like, if I have a reward right next to you as opposed to 10 kilometers away, right? There's a difference. There's discounting there. So what are the consequences of that? There's a lot of things to look at it. And one more thing is, these concepts can you apply in architecture, in design. If you want to build a good apartment, which is comfortable to live, right? You should say, you should ideally have high value everywhere in the apartment. Okay, so can you think of something like that? Apply these concepts to architecture design.
So are you giving scientific?
Basis to.
Feng shui? Is that what you're doing?
Yeah, sorry, basis to.
You're telling us how to compute the value function for feng shui.
For what?
It's the way you use space.
Yeah. Okay.
Wait,
No.
Can I make a question
Yeah.
here? So really you mean state evaluation. In this particular case, the states happen to map
Onto space.
Onto space.
Exactly.
If you were trying to sequence through thoughts, like if you had a series of things that you were thinking about.
Yeah.
First I solved this problem, and I did that, and then all the plants died, and then later than that, right? And you were going through it like a logistical problem. Like the logistics of, suppose you're a warehouse manager, and you have this problem that arises all the time, and you have this sequence of mental images, states that you sequence through and all. You predict that this would be the same sort of problem you have in sequencing through these states.
Yeah, because RL takes care of.
I don't know of any evidence in Parkinson's that
They do sequence.
That have a hard time sequencing.
Yes, they do.
Mental states.
I mean, I don't know the papers right now.
I don't know that.
Sorry?
I just don't happen to know that.
No.
Is that true?
Yeah, it's true. Language and sequence generation. I mean, I don't know.
Language is hard to dissociate the motor part from the sequencing
No, but
of the thought.
I can't tell you the papers right now, but I mean, I kind of know. I can look up and tell you.
So they think slowly.
Oh yeah, but they have a bradyphrenia, for example. I mean, there's a whole name for it. They think slowly, yes.
You know how much more slowly? No, okay.
So so last one, which I'm very fond of, and it can give rise to a lot of discussion. I still remember a discussion we had about 20 years ago about this.
Yeah, I think I stopped it then.
Yeah, I know. Yeah, so the same idea as that is.
I'm not old enough to talk about free will.
So talk about.
Free will.
Yeah.
It's part of philosophy.
I know, correct. Yeah. But I'm stopping short at philosophy. I mean, I'm talking about as a clinical problem. And it's a valid clinical problem, right? So, so first of all, what is willed action? I mean, so in fact, I've seen an interesting paper recently. In clinical literature, neurology talks about willed action, or voluntary movement all the time. In fact, your textbook, your basic neural textbook talks of voluntary and involuntary and blah, blah, blah. But nobody really knows what is voluntary movement, right? I mean, exactly what are the brain substrates and what is the difference in signaling and so on. There's a big article just about that. We don't even know what is voluntary movement. We're using the terms freely. So I'll show you that article. Okay, so if you go to William James, right, the famous American psychologist, he talks about two kinds of voluntary action. One is what he calls idiomotor action, where there's some kind of a plan, like for example a dance movement. You practice it, there's a movement, and it's voluntary, but there's an idea which is shaping your and guiding your movement. Then there is a pure willed action, which is like, okay, I want to lift my hand, and it's not a well practiced, is it a light, and then I'm driving it. Now, the voluntary movement as opposed to sensory driven movement are well distinguished electrophysiologically, because in EEG you can see this long buildup of signal over the midline SMA, which is called a Brett's out potential or a readiness potential, RP. Right. Only thing is and this signal is somewhat weaker in Parkinson conditions, right? And PD people do have trouble initiating movement. Now, although this discovery is very exciting, and it's like, look at the earliest work is from 60s, we still don't have a good theory, actually even any theory, of how is the RP generated, right? What are the underlying mechanisms by which RP gets generated? And if you don't know that, how do you know what is voluntary movement? Because that's what is that's the earmark of a voluntary movement, and you don't know how that signal is generated. And we just can measure it, we see it all the time, right? And it builds up almost like 1 second before the movement onset. Okay, here is the hypothesis. We are saying that there is such a thing called willed signal. I mean, don't ask me, is there free will and all that. I mean, right? So there is a will signal, and it's a weak signal. It needs amplification, right, to get manifest into movement. And that amplification is done by basal ganglia circuitry or through the cortico-basal ganglia loop. So the weak signal, which originates probably in pre-SMA or something like that, one of the highest centers, goes through the cortico-basal ganglia loop, circulates through it, and gets charged up, right, gradually. And this gradual charging up is what you are seeing as a signature in the EEG in the form of RP. And once it reaches a threshold, then you are ready to go, and movement issues out. So how does this amplification occur, and how does basal ganglia amplify the signal? We propose that it happens because of something called stochastic resonance. Now, stochastic resonance is well known in physics, so it works as something like this. Suppose you have a bistable well. I have two valleys here, and imagine you are like a skateboarder or something like that. You are here, and then you need to go over the hill and go to the other side, and you are applying some small force on this on yourself. Like this force applied on you. Unless the force is sufficiently strong, you won't make it and go over to the other side. Now imagine there is a sinusoidal force acting on you, which is very weak, right? By itself, it won't be able to push you to the other side. But if you add to that force a kind of a noisy force, right, which has certain very specific spectral characteristics, certain amplitude, certain spectrum, and so on, then with the addition of that force, even though the actual force is only weak force, it can drive you over to the other side, and you can follow the weak force with a very high probability. This is, in essence, stochastic resonance. Now, what is the connection between stochastic resonance and basal ganglia? Okay, actually there's every connection, because all that you need for stochastic resonance to work is some kind of a gradient descent mechanism, okay, something going down a gradient, and some noise added to it. Now, if you look at our basal ganglia modeling approach all along, right, we don't have gradient descent, but we have gradient ascent, which is just the flip side of it, right? And then because you flip the value function, you get descent, right? So what basal ganglia is doing is helping you climb the value hill, and indirect pathway is adding noise to it all the time. Okay, so the kind of machinery which is necessary for performing RL is also doubling up as a machinery for doing stochastic resonance, right? And therefore you have amplification. So signal goes through this loop and amplifies it, and so that means this noise has to be optimal. So for optimal noise level, so the system goes from first state to the second state and goes over the hill and goes to the other side. If noise is too low, it doesn't kind of move at all. If noise is too high, it moves, but it does something like this. Okay, that kind of, right? So if you look at the three cases, if noise is optimal, right, then so this is like -1 is like the initial state and 1 is like the final state. If noise is optimal, then you give it a small kick, right, and then also add the optimal noise. It goes to the other side and stays there. If noise is too little, then it tries to make a motion and then doesn't get there. That's like your akinetic rigidity. If noise is too much, right? So somebody said, tremor is not a good example. It's more like dyskinesia, so that is fine. But whatever it is, the point is normal basal ganglia seem to be poised on an optimal midpoint, right? Where the noise levels are optimal, and there is amplification, and then there is reliable execution of movement and translation of will into action. But in disease conditions, this noise level could be too little or too high. Right? Too little gives you, like rigidity kind of behaviors. You see all these hypokinetic, right, condition symptoms. Too much, you see hyperkinetic symptoms. So a healthy basal ganglia seems to be poised in between these two. So again, so this is also completely consistent with all other modeling stuff that I talked about. Right? To summarize, we talked of exploration, exploitation, and we said that indirect pathway does exploration, and the whole cortico-basal ganglia loop basically gives you hill climbing over the value landscape. So we did this modeling initially at rate-coded networks, and then we from that we extracted the simplified rule, three-pronged rule of Gen, and then we also constructed more detailed spiking neuron version, right, just to keep the spiking folks happy, because a lot of people think that unless you have a spiking neuron model, it's not a model. So we did also that. But so point is the same model exists at multiple levels of abstraction. At all levels, it tells the same story. Okay, it's totally consistent. And we also did some work on serotonin and basal ganglia joint modeling. And this model, which is work of Pragati, my PhD student, it reconciles the three major ideas about serotonin function. One is it represents risk sensitivity. Doyer says it represents discount factor or time scale of reward prediction. And then punishment sensitivity, which is like the opponency theory. Our theory is consistent with all three of them. All three become special cases of our theory, and this is published. And I think I see that there are a lot of interest in serotonin here, so we can discuss and look at these models. We also did some work on clinical applications, modeling the effect of DBS. So we have a couple of papers there, and then currently we're working on modeling the drug action, because in PD the drug choice is again very empirical. I mean, whether you give L-DOPA or dopamine agonist, it's right. It's a bunch of thumb rules. So if you have a very detailed model where you can simulate drug action, link it to the behavior at the output, right, to be a multi-scale model, then you can consult the model. Once it is very mature and detailed, you can consult the model for your drug choices. So I would like to acknowledge all these people. So my basic story is that, right, we can use computational modeling as a kind of a glue, which can link data from many sources, imaging, behavior, electrophysiology, and you can have common model which will integrate all this stuff. And using such model, you develop treatments for various diseases. First of all, you develop good disease models, right, and then show that it will explain a lot of symptoms of the disease, and then using the model as a base, you have to develop clinical applications. And one long-term dream in the lab, I don't know, whenever I talk about this, student look at me like, what's going on, is to build a whole brain model which is simplified. I'm not the kind of stuff that blue brain guys are doing with supercomputers and all that, but a simplified whole brain model which just has the sensory hierarchies, right, touch, sound, and vision. They combine at some point and give rise to language and space kind of functions. On the other side, you have motor hierarchy, descending motor hierarchies, the musculoskeletal, speech and eye movements, and then between the two you have the decision-making systems in the prefrontal, right, and then you have the three great loops: the cerebellum, basal ganglia, and hippocampus. Right, if you have these elements and build a 1 million neuron model, and that's more than enough, you can really nail down what is essentially going on in the brain, just like a good prototype. And after that, you go crazy and you run it on supercomputers and do biophysical models on neuron and all that. And that you can do more routinely. But without this, I think if you just directly plunge into modeling cortical columns with 35,000 neurons and all that, you're going to go crazy and it's very difficult to becomes intractable. Okay, thank you.
Okay, I have a question about sequencing your thoughts.
Yeah.
So you would predict then in Parkinson's patients that so they would have they should have an inability to initiate thought sequences. They should be bradykinetic.
Or they should get stuck halfway through.
But even to start it, just like they have a hard time starting getting up
Standing
out of a
up,
chair, or they have a hard time starting to walk. They learn all these weird strategies, like they'll sometimes lean.
Yeah, they walk like this.
Yeah.
Yeah.
Yeah, and it keeps
Keep falling off. Yeah. They're falling all the time. Yeah.
That's right. But that's kind of an obvious thing. I was just thinking about sequencing, starting and sequencing through patterns of thought. The only thing I could think of practically was a logistical planner in a warehouse, where you are stereotyped.
Yeah.
You're going through a series of things, and this happens, and that happens.
Yeah.
It's not my impression they all have that that's a subcategory. Is that true?
This, I don't know, I'm not sure.
Okay.
I know they have problem with sequencing, but even reaching sometimes, PD patients, a normal person reaches in one single sweep, but PD patients, sometimes they go like
Yeah.
in stages.
Also, on phenotyping people with the disorder, given your model, it seems that it would be of use to take the model, extract whatever set of free parameters you have, five free parameters.
Yeah, 4 or 5.
Yeah, extract your 5 parameters from a whole bunch of people that have a whole bunch of symptomatology and look at the individual
Yeah,
variation on that. You haven't done
Yeah,
that yet.
I haven't done that, but it's on the cards. I mean, I need collaboration with the clinical people. I mean, a center like this would be great.
Right.
So in fact, I don't have those slides, but basically the one kind of a scenario is, you instead of, you have UPDRS, which is very qualitative, right? I mean, for testing PD. Whereas if you run the patient in the clinic, take some speech samples, handwriting samples, gait, you can get all this data just with a smartphone. You don't need fancy gait lab, right? And then take all this data, invert this data, put it in terms of model terms. You cluster this data. You can get subclassification of all different types
Yeah, I
of PD.
suspect it's a bunch of diseases.
Yeah,
I mean,
yeah,
I
sure,
suspect
sure,
there
sure,
are
sure.
Real subcategories that the models would identify. So.
In fact, there's this interesting article called "There is no Parkinson's disease" by Weber. Basically, he says we don't even know what we're talking about. There's so much of overlap from disease to disease. It's like a continuum, and there's so much of misdiagnosis apparently. I mean, I didn't know that. Like, so.
There's a whole panoply of cognitive symptoms that are sort of poorly characterized.
I guess I'm the only clinical guy here.
Okay.
I'm the neurologist.
Uh-oh.
How do you explain the changes during sleep?
Yeah, so I mean, my vague answer would be sleep and waking is another selection problem. So it's like, just as you select between choices A and B, basically is selecting for you sleep and action, and that is messed up, and therefore you have insomnia. But it's a very vague answer.
I'm not talking about insomnia.
I mean,
Most of the motor symptoms
Yeah.
go away during sleep.
Yeah, okay,
So your rigidity, your tremor, everything goes away during sleep.
okay.
So if your model, obviously, function or structure or neurons are not changing
Yeah.
at the time when you're sleeping. Most of the things go away.
Okay.
Obviously, there is
As
a
those will
different
action.
aspect of sleep problem with REM in Parkinson's, which is involves the sleep physiology. So that is a whole different story.
It'd be nice if there was a way to get them to do willed action in their sleep, like in a dream or something, a well-rehearsed.
No, but how is it?
No, but if they're sitting, they have tremor. If they go to sleep, their tremor goes away.
Right.
Their rigidity goes away. Almost all movement disorder, except 2, goes away during sleep.
What are the two that it doesn't go?
Palatomyoclonus and seizures.
And seizures, right?
Because they are not at all movement disorder. That's why they don't go away.
Right.
So most things which goes through basal ganglia tends to disappear during sleep.
Oh, I'm not aware of that. I'm not sure. I don't
Your
know.
dystonia will go away during sleep.
Oh,
Tremor will go away. Everything, your tone will become normal when you're asleep. If you have bad heart disease, your tone is going to be normal when you're asleep. So if you can just function while asleep, you will be normal. That's the trick you have to.
And what you need is a Parkinsonian person that does lucid dreaming.
yeah.
Oh, they act out their dream quite well, actually,
Yeah.
because they have REM behavior disorder. So they don't disinhibit their motor tone during REM sleep. So they can actually act out their dream.
Wow, it's a very good
That
point.
is- No, they don't have rigidity or they don't have tremor when they act out. They slap their spouse pretty well.
So, can you share some of the literature that explains what is there in sleep and what is there in normal waking? Kind of one.
I mean, I don't think there is a clear explanation.
No, not explanation, but some kind of a review of all these features, just clinical features.
Right, I mean, this is very well known in the
Okay.
movement disorder, that no thing.
Okay.
I'm not a movement disorder guy, so-
Yeah, no, it's okay.
but it is sort of a very well-known feature of human disorders that handles difference.
So what if there's spindle sleep, and what about like skill consolidation during sleep?
I mean, as long as you're stage 2 delta sleep as well as REM sleep, they're all. Obviously, REM sleep you wouldn't have movement disorder because you're usually inhibited, so
Yeah.
You should be all relaxed anyway. But even during stage 2 and delta sleep, so there is a parallel pathway which goes through thalamus.
Yeah.
Which obviously there is interaction because there are 2 loops, 3 loops you're talking about. One is going to the cerebellum, one is the ganglia. And there is a thalamic loop which is not involving the basal ganglia.
Yeah.
All sensory inputs go through thalamus, and then part of them do go through basal ganglia, part of them go directly to the brain.
Yeah.
For example, your visual cortex, there is part which flows through basal ganglia, smart portion. The majority of your visual pathways go through thalamus without any.
And we didn't talk about the hyperdirect pathway from cortex to STN that I've left out.
So there are multiple parallel pathways, and obviously they may be behaving in different ways. Now, visual cues or even sensory inputs changes people's facility. Freezing,
Yeah.
sometimes they touch in a particular
Yeah.
spot
And
and
then
things
you get going. Yeah.
Obviously, visual cues are very
Yeah.
important, clinically used cues. Anybody who's in awakening, putting up
Stripes on the floor.
stripes on the
Yeah.
floor changes your walking. And that is obviously, it can to some degree go no-go, maybe
Yeah.
visual cues. And those visual cues need to be very high contrast.
Hmm.
So you put the shades of pink, it will not work.
Yeah.
It has to be very high contrast. And obviously contrast is also a feature which Parkinson's don't get well because of retinal deficit of dopamine. Retina has a lot of dopamine. So first thing they lose actually with smell is the first
Yeah.
thing. Another thing they lose is the ability to distinguish contrast.
You lose chromatic contrast
Chromatic
without
contrast. That happened very
the
early with Parkinson's. Early Parkinson's they already have. But I don't think that is the reason why they have it.
Yeah,
So
yeah.
the sensory processing is probably more of an issue which helps them. And I don't think the freezing is very typical during going through the door. I mean, it's a classic feature. But freezing do happen
Many other places. Yeah, yeah. Correct.
no matter where they
There's
are.
turning and any trigger, yeah, anything can trigger.
Freeze, and they can move, and the next moment they move fine.
Yeah.
And I have a colleague who's a neurologist who has had Parkinson's disease, Parkinsonian syndrome. But he would freeze at one moment and then
Yeah.
he'll be running next moment.
No more
Okay.
questions. Well, let's thank the speaker again.